Vanakkam. Welcome to this course on classics in neuroscience. In this set of videos, we have been looking at neural circuits in the spinal cord, retina and invertebrate systems. We have looked at the spinal cord in the past. For the last couple of videos, we have been looking at the retina, and this video will continue our discussion on the circuits within the retina and the crucial seminal work of Stephen Kuffler and Horace Barlow to improve our understanding of retinal processing of the visual system. Stephen Kuffler famously collaborated with Eccles on the neuromuscular junction, their famous work with cats in Australia. He continued working on other topics, for example, on the contrast enhancement in the mammalian retina. Remember, the previous work was on Limulus, on crab by Hartline. Here he extended this work to the mammalian retina, and which he published on the discharge patterns and functional organization of the mammalian retina as a paper. In collaboration with his colleagues and friends, he invented, for example, with S.A. Talbot, he invented a new ophthalmoscope that could provide specific stimulus to the mammalian species. So, in this ophthalmoscope, it was possible for Kuffler and his team to stimulate two different points, one light and another dark, or both light, for example. So, he could study the response to more than one stimulus that is happening simultaneously. It was an inventive idea. Also, he invented a method to record from the in vivo cat retina by inserting an electrode. So, this metal microelectrode was inserted at the edge of the eyeball. Yes. So, he found that in his results, he found that these cells responded primarily to contrast and to moving stimuli rather than diffuse light. It is very important that these cells respond to moving stimuli, a fundamentally critical evolutionary purpose. You need to be able to catch the moving prey. They used to study this in cats. So, obviously, cats are very alert to stimuli that are moving relatively fast, for example, the mice, which is their food. So, this image of the paper of a micropipette on a retinal ganglion cell surrounded by two concentric circles, one which is an excitatory center, another which is an inhibitory center surround. This stood as the beacon of this new line of neurophysiology, electrophysiology on the visual system. This was a absolutely crucial, critical advancement made by Stephen Kuffler. So, Stephen Kuffler was an extraordinary scientist who was suggesting many inventive ways and elegant experimental apparatuses and elegant experimental systems and model systems to study. Students were attracted to work with Stephen Kuffler in a very big way. Some important students, some important members of his team, David Hubel, Torsten Weisel, about whom we will discuss in the later set of videos. For example, David Hubel and Torsten Weisel were people who collaborated with Stephen Kuffler and benefited from that collaboration in a very big way, who went on to win the Nobel Prize for their work on the visual system. Do check them out. Then we move on to the work of Horace Barlow, who could trace his genealogy back to Charles Darwin as the great grandson of Charles Darwin. He published a paper on frog retina work on the stimulation and inhibition in frog retina, and he worked on the principle that there is convergence and there is overlap between the neurons that were responsible for this senses, that is the visual system. And he discovered that there was inhibition of light outside the receptive field of fibers that were called on-off fibers that just detected presence or absence of light. Also, he found that a white spot that is surrounded by black appeared brighter than the same white spot that was surrounded by grey, that is a form of contrast enhancement, and he could explain this using his physiological recordings. Kuffler and Barlow's observations essentially suggested that both inhibition and excitation in the neuron's receptive field were responsible for the perception as a whole. So, that is, you cannot include only excitation, both inhibition as well as excitation were to be included as part of the receptive field of a neuron, and it was Horace Barlow's experiments that provided the first evidence to suggest that there was this movement sensitivity for visual cells. With this, we come to the end of this video. In this video, we looked at the work of Stephen Kuffler and Horace Barlow on contrast enhancement, movement sensitivity and other such things in the retina. Thank you for your attention.